Tomorrow lives in the future. It's intangible. Tomorrow is what's missing today. It's the promise of new, the unknown, the real life we haven't experienced yet. Tomorrow is a world where we understand the biology of cancer, where food sustainability isn't a what if. Tomorrow is the assurance that when you get sick, you're taking the right medicine. It's understanding your ancestry so that you can appreciate those who came before you. Tomorrow is where human health is different. It's better than it is today. Tomorrow isn't distant. It's closer than you think. It's a world where your research doesn't just dwell in journals. It reverberates in the heartbeats of those you touch. The concept of tomorrow is no longer elusive. It's the reality we're creating. It's the tools you need to see the why so that we can change the what. Sequencing technology with the accuracy, precision, and scale you've dreamt of. Available now. Welcome to tomorrow.
Good afternoon, and thanks everybody to joining us right after the lunch session. As you can probably tell, I'm not Jonas Korlach. Jonas, unfortunately, fell ill and was not able to join us this PAG. So he sends his regards. The plant and animal community is always very close to his heart. And with that, my name is Jacob Brandenburg. I'm the plant and animal sequencing specialist here for PacBio on the marketing side. And I'm going to be giving you the introductory remarks today. We have an exciting list of speakers for you, so you can take a look here. So we have Doreen Ware, who's going to be speaking on some of the work generated out of our new Kinnex RNA kit. And then we're going to have Ethan from Zymo give you an update on Kinnex full-length 16S. We're going to have Carolyn Howard give an update from a lot of the exciting work coming out of the Wellcome Sanger Institute and some of the exciting milestones they've achieved this year. And we'll be rounded out with Josh Clevenger with some of his work on low-pass sequencing utilizing the new Revio system. So last year at PAG, we announced the Revio system itself, and those systems started shipping in January. So about 10 months, and over the last 10 months, we've had customers across the globe placing these systems in their labs, and not just placing these systems, but also utilizing them. So we're starting to see the gigabases roll out and quickly surpass the existing Sequel IIe fleet. So after about 8 months, the Revio systems that have been placed in 2023 surpassed the gigabases that have been generated by all the Sequel IIe systems out in the field. And as of a December benchmark, we actually hit 16 petabases worth of sequencing data in just a year's time frame. So again, to give you some context, one Revio system is equal to about 15 Sequel II in terms of output. We've also started to see the first publications rolling off the press. So a lot in the variant calling and precision space in humans. So you can see some of the ones here that were highlighted at ASHG. And on the plant side, the first one that we're seeing utilizing our new system was out of the International Weed Genomics. And when we say weed here, we mean invasive species, not the cannabis side. And it didn't just stop there. We also saw a multitude of other organisms that utilize the Revio system in the publication space. So anywhere from dragonfly to ornament fish and different insects and plants. And the Wellcome Sanger Institute has been putting out different genomes each week for the last couple of months. And we'll hear Carolyn talk more about that upcoming. We also now have onboarded a lot of end-to-end workflows to support our Revio system. So we purchased Circulomics and brought their nanobind technology into our portfolio. So we have DNA isolation capabilities here at PacBio. We've set up a nice PacBio compatible partnership program to help with the shearing and the library prep and the automation side. So you can see some of the different companies we work here with in the automation space. And finally, on the sequencing side, now with the Revio system, customers are able to sequence 4 Revio SMRT cells in a single day, and that's enough to do a 3 gigabase genome per SMRT cell. So you can actually sequence 4 at 30x coverage in a single day. And we're also coming out with a lot more of the secondary analysis tools. So you can see a smattering up here of ones that have also come out this year. A lot focusing on WGS and structural variant space. We also have cloud workflows if you're interested in the informatics, and we also have a lot of GitHub and how to do the sequencing and analysis for our iso-seq RNA and also metagenomic pipelines. You may have seen the PR press release this morning. We're announcing our new nanobind pan-DNA kit. So this is actually a consolidation of our extraction portfolios. So currently we had 3 different kits on market depending on the organism you were extracting from. And we are excited to announce that now this can all be done with a single kit, and we've onboarded additional organisms. So now we fully support insects into the pan-DNA kit. And not just that, but we've also expanded our high throughput capabilities for extraction. So we were always able to do mammalian cell and human blood in a 96 plate format. And with the announcement that went out this morning, we're going to be expanding that capability for mammalian blood and also non-animal blood, as well as gram-positive and gram-negative bacteria. You can see some of the automation that this high throughput extraction is done on. So the Thermo Fisher Kingfisher Plex, and also the Hamilton Nimbus Presto. And if you want to learn more about this, since it is right off the press, we have a poster that's being presented at the 3 to 4:30 session. So that's poster number 0633. And our extraction team would be happy to talk with you more about these exciting announcements. We've also gone and validated different DNA shearing methods to support high throughput. So these are shearing methods that you can do in a 96 plate format, and we really have 2 different methodologies that we employ. So the first is plate-based shearing. This was developed on the FastPrep 96 and also the GenoGrinder and Mini-G. And we have pipette shearing that we can do on the Hamilton NGS Star and also the Hamilton Nimbus as well. If you take a look at the graph on the right side, you can see how these shearing methodologies compare to the Megaruptor 3, which we use as our historical benchmark. And again, we're very happy with the peak sizes that we see coming out of all of these methodologies. We also just released our first ever software upgrade for the Revio system. And this one was important to us for a multitude of reasons. So first and foremost, it really enabled a lot of additional applications, and you can see the ones listed there. The reason for that enablement is we are now able to sequence all insert sizes. So when the Revio system launched, you couldn't sequence under 3 kb. That still had to be done on the Sequel 2 iteration. And so again, we're happy to announce that with the same polymerase and library prep kits, we can now sequence all fragment sizes on the Revio system. We have also made it more streamlined. So we have a pre-run feature as well as adaptive loading, which I know the customer base has been asking about for years. So again, happy to finally have that delivered. And then finally, this new Revio software update is also really enabling the Kinnex kits that we're going to spend the majority of the time talking about now. So here's a look at the list of applications that was enabled with this new Revio update. And you can see the Kinnex kits here circled in pink. So we have 3 different Kinnex workflows. We have our single-cell RNA that was already previously accessible. We have full-length RNA, so this is our iso-seq protocol that is now utilizing our Kinnex kits. And finally, we have full-length 16S sequencing. So let's dig into the Kinnex RNA sequencing side. So I'm sure a lot of you are familiar with our standard iso-seq protocol. So again, this is full-length isoform sequencing. And again, this is a smattering of publications utilizing iso-seq over various organisms that were published last year. And our iso-seq has always started at the gene level and given you full-length isoforms. So you can now look at different splice variances and different isoforms related to a single gene. What the Kinnex kit does is it takes each one of these isoforms, and then it allows us to concatenate multiple isoforms together, and that will form a single HiFi read. So a HiFi read on average is about 15 to 20 kb in length, and traditional isoforms in the plant and animal space tend to be anywhere from 1 to 3 kb. So there's a lot of wasted real estate on a HiFi read itself, and concatenating multiple molecules together allows us to increase the throughput of those isoforms and 16S and single-cell reads with this single methodology. And when we pair that with the Revio system, we expect about 40 million full-length non-chimera reads per Revio cell. This is a 16S increase over the Sequel IIe iso-seq methodology. And the Kinnex kits themselves have enough to do 12 reactions, and they give you the flexibility to do additional barcoding, so you can do up to 48 unique cDNAs per Revio cell now. We also think that this is going to increase our recommendations for what we consider a gold standard annotation. So you can see here on the graph at the bottom that we start seeing saturation at about 10 million reads. So at 10 million reads, we're hitting about 80% of known isoforms for the different species that are listed here. And we think that that's a pretty good benchmark where we're starting to see that leveling off effect, where going higher, you're not getting much more gains. We also wanted to show that we weren't impacting transcript sizes, so that we are still able to capture those longer transcripts as well. So if you take a look at the graph up on the top here, you can see that for all organisms listed, we still have that right tail for those longer transcripts that we know exist within an organism's biology. If you were at ASHG, you may have seen Andrew Sturkakis and from University of Washington, as well as Jonas Korlach, speak on an initiative they did where they were able to sequence 4 high-quality omes on a single Revio cell. So this is the genome, the methylome, the chromatin epigenome, which Andrew's group has a specific methodology that helps you assess open and closed chromatin structure, so they call this FiberSeq, and then now the transcriptome with Kinnex. So what they did is they did that FiberSeq reaction, and then they went through the normal genomic DNA prep, or the standard HiFi prep. And then they also took RNA, went through this Kinnex workflow, and they pooled these 2 libraries together in a ratio of 15 parts to 1 part for the Kinnex. And what they were able to show here is that with all the analysis tools you can see on the right, when they went to visualize the HiFi data, this is what they see. So I know this looks a little overwhelming and messy, but you can see here that they're phasing the 2 haplotypes. They're also getting that CpG methylation that you see up at the top. They're now being able to assess that chromatin epigenome in purple in the middle there. And then finally, the transcriptome is matching up, and you can see that correlating there at the bottom. This was originally done on human, but there's no reason this can't be expanded out into other organisms. So Andrew Sturkakis' group is happy and willing to implore this across the tree of life. And so if anybody's interested in this methodology, please see us at the booth, and I'd be happy to talk more about 4 unique omes in a single Revio cell. We've also been working very hard to create a lot more tools on the bioinformatics side to help make this data meaningful. And you can see here that a lot of this is in the structural variation space. And so we can now call quite a few different structural variations, including tandem repeats, segmental duplications, as well as SNVs. And then you see that methylation calling down there at the bottom. So this is all in the WGS workflow. Again, we also have an iso-seq.how that walks through all of the RNA tools we have available, and there are quite a few metagenomic tools that we'll get into in a little bit here. So next I wanted to talk about Kinnex 16S. So again, this same concatenation methodology can be applied to other amplicons of interest. So on the 16S side, we use the same concatenation, but instead of an 8x concatenation on the RNA long read side, we're doing a 12x concatenation. So what that means is you're able to get up to 1536, or 4 different plates of 384, samples multiplexed together on a single Revio cell. And this is for full-length 16S sequencing. So now on the Revio system, you can expect outputs of about 60 million reads per Revio cell. And this brings us in line, if not cheaper, than the short read technologies. So our hope here is that rather than people looking at just the V3 to V4 region, that they're going to be able to get much better strain and species level resolution by looking at that entire V1 to V9 region. And the cost is going to be very similar to what people are expecting in the short read side. And then if you take a look down at the data analysis, there are a lot of different tools that you're welcome to implore to get at that species and strain level information for this workflow. So now with the addition of long-read RNA and 16S, you can see that this is our Kinnex workflow. So the only real difference here is the level of concatenation. So single cell uses 16X concatenation, 16S uses that 12X, and then long-read RNA uses 8X. Otherwise, it's the same workflow for the different amplicon sizes. So I also wanted to show this trend in genomics that Jonas spoke about last PAG. So Jonas was very excited to kind of show where NGS started across the different omes that you see listed on the left here, and where NGS is able to get us to today, and where he was excited about the Revio system helping customers expand into the future. So you can see that while we're starting to get a nice picture of a lot of these different omes, there's still a lot of collapsed haplotypes, gaps in assemblies, as well as missing species and strain level identification. And we're happy to announce that in a single year on the Revio system, we have really come out with products to address all of these missing components from the NGS workflows. So on the genome size, we have now a very complete variant color pipeline for whatever types of variants you might be interested in. We have the Kinnex full-length RNA kits to assess that transcriptome, now at scale with that 40 million reads per Revio cell. We've enabled the 5MC calling in the CG context for epigenetics. We can start assessing the cell atlas again with Kinnex single cell workflows. And then finally, we're going to get that species and strain level identification with Kinnex 16S, as well as a lot of the new MAG pipelines that the team has worked hard to develop. And I didn't want to leave out our short-read sequencing platform. So if you recall from last year as well, we announced our short-read capabilities. Again, the differentiator for us here is that this is really a Q40 plus for the majority of the reads coming off of the short-read side. And this system has formally been launched and is shipping globally to different countries. And you can see some of the nice quotes here from some of the customers that have already gotten their hands on this data. They go on to make very nice comments on how they're already utilizing this and that they're actually sometimes seeing the majority of the reads being even in that QV50 benchmark. We also announced an acquisition this year of Apton, and so this acquisition is going to allow us to scale this Onso technology into a much higher throughput machine, and hopefully we can really expedite the time it's going to take us to get there with this acquisition. So what's in development, it was announced at JPM this year, is that we're hoping to get to billions of reads with the runtime and metrics that you see here, and again, this is all based off of that Onso technology that we presented last year. I also wanted to remind everybody that registered for the party. First of all, thank you. We're excited to see everybody. Just a quick reminder that you will need to show on your phone, or if you have printed it, just confirmation that you have registered to get on the buses. And the buses will be picking everybody up right across from the registration desk right outside here. So right at 6:30 sharp is when we're hoping to start shipping those buses off to the aquarium. So please be there in a timely manner if you are planning to attend. And I did want to apologize. The entire aquarium is open for the event tonight, except the penguins. It's past their bedtime. So we did not take that into account when we were originally planning this. But yes, so we won't be able to see the penguins. They will be happily sleeping at that time. And with that, I'm happy to yield over to our great list of speakers here. So again, thank you for the workshop. We will be having a Q&A session at the end, time permitting. So if you could please hold your questions for the speakers until all four have gone, and then we're going to be more than happy to open up the floor. And we have microphones on either side here. So if you could just form an orderly line at the end of Josh's talk, we'd be happy to take questions with the remaining time. And with that, I'm going to hand it over to Doreen Ware, and she's going to talk about her Kinnex RNA work.
All right, I'd like to thank all of you who are here to listen to the talk, and as well as PacBio for the opportunity to present the work and the long-term collaborations that we've had. So what I'm going to do is break my talk down into 4 sections. I'm going to be giving you big high-level background. I'm going to be talking about some of the recent work on Revio that we've done with some sorghum reference genomes. I'm going to talk to you about the characterization of some new isoforms using that Kinnex kit that we've just heard about. And then I'm going to give you just a sneak preview. It's very early on of looking at peptide spectra to actually validate some of those new isoforms. So it's getting hot, right? All of you who are here know that climate is changing, the erratic things that we see, as well as it's getting hotter. And so when we're starting to think about that, we need to think about either adapting our existing crops or maybe reusing some of those crops. And so looking here, this is the FAO stats on cereals, and you'll see that there's been an increase in production in maize, rice, and wheat. But I'm going to be talking to you about one of the commodity groups, sorghum, that I work actually closely with in this country. So many of you are probably aware that the U.S. is the largest producer of corn, but many of you may not have been aware that the U.S. is the largest producer of sorghum. This means that we actually have a commodity group and a cereal in place here that actually can be very climate resilient. And we can think about how to work with that and how to expand that. And so when we ask the question, why are we sweet on sorghum? There's lots of reasons. It's because it really can be cultivated in many locations. It's very heat tolerant, it's drought tolerant. It actually can be grown in tropical conditions as well. And it has a lot of industrial examples in addition to basically being food security and for animal stock. Oh, and I just want to highlight in that picture there, what you're seeing is this is coming from a report done in Europe in 2011 on looking at sorghum as a new potential forage, and what you see there in brown is actually corn. And so that's the stay green phenotype that you're looking at. So high level, technologies continue to improve, and so whether it's the way that we basically collect data on phenotypes or whether we look at the genomes themselves, I've been doing this for more than 20 years, and in 18 years the price of the genome that I spent a lifetime on was $30 million, and now it's $3K, okay? So this opens up a tremendous amount of opportunities. And then along the way with the different sequencing technologies, there was integration of different technologies to give you that high quality. Everything from the traditional Sanger, then we went to short read, then we went to integration of optical maps, then we went into the opportunity to do long read sequencing. And really what we've been doing over the last 4 or 5 months is leveraging the new Revio technology that you heard with to actually bring and address some of the additional genomes. And we've been working directly with the sorghum commodity group and a working group in identifying what are the priority lines. These actually have to do with wild varieties or existing land races that may have quality traits in it, as well as their elite lines where we want to basically really characterize what's in them, both good and the deleterious alleles. So one of our approaches to think about, can we just do this with HiFi alone, right? Because it makes it a lot easier if we don't have to integrate it. And so our target was to really look at 30x coverage, the HiFiism assembly, scaffolding directly to existing high-quality reference genomes, and then doing a double check there. So we've been working directly with Brian Scheffler's group, who had access to a new Revio in September, and this is really hot off the press. We had about 14 genomes. I'm showing you 10 here. All of them, except for 2, had N50s above 50 megabases, so these were really good quality. When we look at the BUSCO scores, these are just straight preliminary assemblies. We get BUSCO scores of 98%. So these are suggesting that with just the HiFi alone that we can do this. So for scaffold assemblies, this is a first example of a seed parent in the hybrid, and you can see that this is really nice. We have a total of 13 gaps and 13 gaps total, and the largest one is 46 kb. So we actually feel that we're at the point now where we can turn these around pretty rapidly. It takes more time to grow up the tissue isolate the DNA and make the library than it does for us to do the sequence. I just heard that I could do three genomes a day, not just one, right? And so we can do that, and we can assemble in a day. So this is a real game changer. Okay, as I mentioned, my group has been engaged in stewardship of these genomes for a long time, working with EBI on the Grameen side, as well as Oregon State University and recently Penn State on the CLIM tools. And we host more than 250 genomes in there. But over the last 4 years, we've really been focusing on the pan-genome sites and working with 4 commodity groups: rice, grapevine, sorghum, and maize specifically. And so in working with those, I call this the good, the bad, and the ugly. So on top in yellow is the annotation of those genomes, and with the ability to create other groups and no longer sequencing being located in one center, many groups can produce these genomes and work on them. So you'll see that there's heterogeneous differences in the number, but I want you to look at the mean gene length in gray, and you'll see there's differences in size. These have to do with annotation workflows and potentially the quality of the assemblies as well. While the genomes are coming along, the genome annotations are still hard. So I just want to give you a history of the annotation. So we often think about the history of the sequencing, the cost. But the history of the annotation, I can tell you that first B73 genome took us almost 2 years to annotate. If we really looked at the time, it was very hand-intensive and laborious. Then we went to newer approaches in the next round that took us about 9 months. I would say the version 4 is when we introduced the full-length transcripts, and it took us about 3 to 4 months to really annotate it. And then with the most recent version 5 first pass, once we agreed upon how we were going to annotate it, it took us about 2 weeks to run an annotation. Now I'm telling you we can get 3 genomes a day, but it's taking 2 weeks to run an annotation. That is problematic. And so one of the things we've really been looking at is how can we move forward to improve that annotation. And so the approach that we're basically taking is what we're going to call producing a sorghum pan gene set. And from this, we basically are taking a representative, what we think is the best model from each class. So the core genes that have in every one of those accession, and then one representative from each one of the genes that are coming from there. And you can see as you get closer to the shell, it's mostly, in this case, sorghum-specific genes that are in there. And so we're not sure if these are really newly evolving genes or they're just garbage that showed up, but we're including a version of those, because we can actually validate them with some of the transcript information that we're looking at here. So we use a workflow where we're basically building these orthologs groups, and then Andrew and Kapil and Sharon have set up, and Genuine, a set of filters that we can then select the representative model. We prefer to take the representative model from BTX 623, which is what the community has used, and we actually think it has the best set of annotations. And then we take from representative models from the other genome when it's there, because believe it or not, I can tell you that B73 has pseudogenes that are basically loss of function based on genetic variation that are real. And so we need to go to the next one. So then we can take those models and we can use liftoff then to propagate them rapidly to these new genomes that are coming up, and then refine them, and then we can give them an annotation edit distance using Maker on what the score is. And we did our first pass of this with the carbon partitioning nested association population in sorghum. We get about 41K genes on average in these. So one of the real limitations, though, on that is actually the accurate full lengths and isoforms of full lengths. And so I had an opportunity to work with Liz when they were in the development of that and provide her some RNA samples that the kits use. Now, I'm putting this slide up here because I want to ask you guys, do these plants look stressed? All right, so they're grown as seedlings, and we germinate them, where we wrap them in paper towels, and before we germinate them, we sterilize them. So we beat them up and put them in bleach and expose them to other things, right? Then we basically wrap them up in these paper towels and then grow them. And here's the seedlings that you're looking at. So based on those data, the RNA that we provided from these samples, this is what we get, okay? So we see examples of exon skipping, highlighted in green, intron retention, highlighted in the red box, and then 5' and 3' alternative stop sites. So this is the data. This is the data. All right? We know in plants that when you stress them, you have an increase in intron retention. We can ask questions of what that's involved in. What was really nice here is with the 10-day-old, these were 10-day-old seedlings that we used for the RNA. We ended up touching, I should have said 65% of the known loci, not the known transcripts. So we had 65% of the known loci in there. We end up getting many more isoforms, antisense transcripts, and novel ones in there as well. I'm going to focus on the isoform work, but we really find that the depth of this data is really setting us up to actually start to look at using these features and training them to build better models of the annotation. So the first thing I want to say is that if you just took the existing monomer kit, run on a sequel, and you compared it to the Revio kit and the Kinnex, we get an 18-fold increase in the final collapse forms. That's really a game changer for us as far as getting things. If we take a look at that as just comparisons between the monomer and the Kinnex, we see an 18 to 22-fold increase in the FLNC reads, and we get a 9-fold increase in the collapsed isoforms. All right, so then the Kinnex research and development team processed these for us through their workflow, and you can assign various structures associated with these form matches. And what I really want to focus on here is the category that are novel isoforms with a new combination or novel isoforms with at least a new splice site. And so it's showing you that we're seeing many of these. And if you look at the blue, that bar represents the Kinnex and the Revio there. And the yellow and that you see there, that you don't really see there, shows you the isoforms that we see if we were just using the normal monomer on a sequel now. So it's showing you the amount of diversity that we're seeing. So do we see intron events? Yes, we do. So we see a lot of intron retention here. Now that's why I asked you, are those plants stressed? I don't know. Maybe they are, probably are. We see a lot of intron events. We see some of those intron events are maintained in high frequency in the transcript. So these are just not one-offs. And you saw examples of that when I was showing you that image. I think image is always 1,000 words there, and in different structural categories. So when you look at this, though, so what I really want to point out, when you look at those examples, you see a shift in the transcript size. And those shifts in the transcript size are likely due to the intron retentions that are happening there. So we do see an increase in the overall length, and these may be involved in the intron retentions that we see. So intron retentions and alternative splicing, this is really nice. If you haven't read this, this is a nice review that was recently done in the Plant Cell. And really we have different transcript fates. So it could be, these transcripts could go to NMD. They could be involved in nuclear retention and somehow involved in refining the transcript's regulation. They could be translated. So one of the questions we wanted to ask is, can we identify proteins associated with these novel transcripts? So I was like, oh, yeah, we can do this, we can do this. So I went over to our mass spec guy and I said, hey, Paulo, can you work with us to see if we can identify any of these new isoforms? And he said, yes, I can. And so what we did is we used those isoforms to build a proteogenomic database. And then, so what you see in red is the proteome that would come from those new isoforms. And then you would see on the left-hand side in black represents the isoforms that would come from the UniProt database that's based on BTX623. And what you can see is the overall residuals from those are the same, so there wasn't a huge skew in the amino acids. So what we did this time is instead of giving the whole sample, we divided this up into leaf stem, root top, root mid, and the root apical meristem. And so what you can see here is the spectra that's coming from that. And so the spectra were matched to the theoretical peptides from both the UniProt database and the big database that we generated. And so here's really where we get to the interesting part. So everything in gray is what's common between those databases and we're seeing in the spectra. And what you see in black and red are what is unique to those databases and we're seeing in the protein spectra. So what we see is we do see examples of some of these novel isoforms in the protein spectra. And so if we look at the isoform counts associated with the protein spectra, this is what's considered associated with those full splice matches. So that's like the reference genome. But we do see other examples associated with these nice novel isoforms. And we can also take a look very specifically at intron retention. Excuse me. And so what we see is we do see examples of intron retentions, and I am not a mass spec person, but the person who runs the proteome facility tells us that these are really showing strong experimental evidence based on the fragmented spectra and the ions there. And so this shows you an example where we can start to pick up some of these non-canonical isoforms. So what I want to leave you with is that we are really in great shape to produce new assemblies, get at genetic diversity that could really be important for breeding as we move forward. You know, from a DNA library to a sequence in an assembly, is 3 days now. And based on the annotation workflow, good or bad, we could say 4 days to annotations. We know that the gene annotations definitely need work. We think that access to more transcripts will definitely help this. And then we also have the opportunity with these new kits to actually ask some detailed questions about what happens. So I'm interested, what happens to pollen when it's 10 degrees higher? Do I see shifts? What happens in grain during grain filling when we have a freezing or 10 degree? So we have an opportunity to start really taking a look at that. And then the other thing, it's just a very preliminary work, but it says some of those isoforms that we see that we don't really like, we would rather not have intron retentions, are actually there. And so this suggests that we might have more diversity in the existing proteins than what we've seen before. But overall, most of the protein spectra is associated with the canonical, but there is more diversity out there. So I'd like to thank the people who are involved in this. So very specifically, Nicholas Gladman, a new scientist, USDA scientist, was spearheading the priority of the germplasm in the group. Kapil and Genuine Andrew were associated with the annotation workflows, and Audra and Michael handling the samples. Paulo Schifano was the proteomics person, and we couldn't have done the reference assemblies that we're really moving forward with and working with the Sorghum community to get these resources out without Brian Scheffler and his group down in Stoneville. And then we're very excited to see the data that has come out of the Kinnex kit. It really shows promise for the future. And we thank our Sorghum community for the rankings they've given us and then access to the computer resources. And most of all, I'd like to thank my whole group because they do all the work. And if any of you are interested in more information on genetic variation, we have a workshop and we're going to be talking about RSIDs and how we should be using those for the agricultural genomes. They're a standard in human, and I encourage you to come.
Great job. Perfect job.
Do you want me to hit?
Yeah. I can also do it if you want. Great, thank you so much, Doreen. Really impressive how much you guys were able to accomplish in such a short time. So kudos again to you and your group. Just a reminder, if anybody's standing in the back, no pressure, but there are a lot of seats open up in the front if you guys want to kind of work your way this way. Otherwise, without further ado, it's my pleasure to introduce the next speaker. So we have Ethan Tai from Zymo, and he's going to be giving you an update on what they're able to offer and the results that they're seeing with the Kinnex full-length 16S kit. So take it away, Ethan.
Hi, everyone. How are you guys doing? Do you have a time to enjoy lunch in the Nice weather in San Diego. Great, great. Glad to hear that. All right, before I start my talk, I just have one question for all of you. Have you ever wished that you have more than 24 hours a day to enjoy time with your family, to read more paper, to design your experiment, to analyze the data? If the answer is yes, I have a solution for you. So my name is Ethan again. I'm from Zymo Research. Just a quick background about Zymo. So we established in 1994. Our headquarters in Irvine. We have established our global presence around, well, all continents, in Germany, Brazil, China. We are an innovation-centric company that have a very strong R&D capability and IP portfolio. We also have a strong distributor network for more than 80 partners. So throughout the timeline at Zymo, we develop a lot of kits and use those kits to implement application. Some of the highlight will be in 2015, we launched a patent Zymo Pure technology for high-yield plasmid purification. And 2016, we launched a Zymo Biomics pipeline for consolidated microbiomic. So at Zymo, we do a lot of things, but I'm here to actually talk how we actually help the community to work with microbiome. So we have a complete portfolio for microbial solution from collection to conclusion. We have microbial standard, sample collection device focused on microbiome profiling, preservation, DNA RNA extraction, especially for long-read sequencing, library prep, and of course, NGS analysis. And with all of this, we actually combine it to provide a solution for you when you need more time to do the thing that you want to do. So have you ever sent a sample to a service provider and wonder, what happened to my sample? How did you handle it? So I just go through a very quick tour of how we handle your sample at Zymo. So when the sample comes in, depends on the condition. Is it frozen or is it in the RNA shield? We will have to establish the condition of sample and actually establish how we're going to process sample. Do you need, is a soil sample or fecal sample? Does it need to remove PCR inhibitor for downstream application? Next up, we do DNA RNA extraction using our high-throughput automation platform. And then we also carry that high-throughput automation platform to actually provide a high-throughput library preparation. Library then QC and sequence on either PacBio sequencer or Illumina platform. But bioinformatic analysis are done and QC thoroughly for bioinformatic team to ensure the most accurate result to be delivered. So we combine all of this high-tech, state-of-art workflow to provide a full-length 16S sequencing, which launched last year in May. So the idea of service is to simplify your research, to give you back the important factor, which is your time for your discovery, for your time with your family. So we know that full-length 16S actually provide a better picture looking at the entire 16S genes. So I just want to quickly capture the advantage of use full-length 16S. So with full-length 16S, again, since we were able to sequencing analysis of the entire 1.6 kb of the microbial 16S genes, we now have a more complete picture and able to achieve unrivaled species resolution. With 16S, it's more affordable than shotgun metagenomic sequencing to achieve the same taxonomy resolution with a quicker turnaround time. And because it's an amplicon-based sequencing, now you can apply this technique for a wider range of sample type. So currently, the most popular method for full-length 16S analysis is not monomer amplicon sequencing, but this process has some technical challenges. As a service provider, we receive a lot of different type of sample, and we need to pull them together for a more effectively sequencing. Because of the variety of condition and input, we have a difficulty for read normalization, which limit the number of sample we can put in one SMRT Cell. And because of that, we cannot utilize the full capacity, and was not as affordable as short-read 16S. Into Kinnex 16S RNA application. So this aims to maximize the advantage of HiFi read length up to 20 kb. The approach is based on the MAT-seq method to concatenate multiple full-length 16S amplicon into a 20 kb molecule. So these 20 kb molecules are sequenced after read segmentation. We end up with multiple copies of the same 16S amplicon. So with the preliminary data using fecal reference standard, the data output showed that a significantly increased from 2.6 million HiFi read to a whopping 30 million HiFi read. So at Zymo, we are very excited of this improvement. To further testing the robustness of the technology, we send PacBio extracted DNA plate with a diverse sample type and condition. This is to mimic these real sample condition my service team handle on a weekly basis. So as you can see, we send condition, as in DNA concentration, range from high biomass, high concentrated DNA to low concentrated DNA. Host DNA present, such as human, animal, plant. A wide range of microbial diversity from low diversity to high diversity, such as fecal, and the PCR inhibitor present in these sample. And of course, we also include various microbial standards because great science requires controls. So these samples are light. On a Voltec for 40 minutes at max speed, the DNA is purified using Zymo Biomic DNA extraction kit. So afterward, the sample were sent to PacBio for library prep and sequencing. The data came out much better than we expected. Comparing Kinnex 16S and monomer 16S, the HiFi read output is as well as the average read per sample increased more than 10 times, as shown on the table. So when we compare the read output of fecal and soil sample, we see a tremendous difference between two approaches. Keep in mind, these library prep process can be further optimized for better normalization. However, when you look at the fecal sample, Kinnex 16S versus monomer, there's a 10-time read increased output, while soil sample is double the amount of read output. Profile-wise, when you take a quick look at these profile, the top part is the Kinnex 16S, and the lower part is the monomer 16S. With a quick glance, there's not much of difference, but when you look at the unique sequencing, Kinnex 16S have more than twice of the unique sequencing in fecal sample and 7 times more than in soil sample. So, however, just like any other application, metagenomic workflow has a unique set of challenges, especially for long-read application like full-length 16S. So when you're collecting your sample, we need to be confident that the composition of these samples is not changed between the point of sampling and DNA isolation, because this could impact the overall outcome of your study. We also need to preserve DNA integrity to ensure the sufficient quality for sequencing. When isolating DNA, we must ensure the uniform lysis of the various microbes present to ensure that we obtain unbiased profile. And for long-read sequencing, of course, DNA length is another key consideration. For library prep, the workflow still have multiple steps, which increase hands-on time. The workflow is need to be optimized to be further streamlined with different sample type, especially for a service provider. And of course, as important as sample prep and sequencing, downstream bioinformatic analysis require a robust pipeline with database free of sequencing and taxonomy error for accurate analysis and interpretation. As two pioneer companies in the microbiome research, Zymo and PacBio are tackling the challenge together on a different front. As PacBio keep pushing the boundary of sequencing technology with newly launched Kinnex 16S kit, they have successfully increased the sequencing throughput as a new technology allow multiplex thousands of samples to be sequenced at the same time while maintaining the high read output with high accuracy. On Zymo's side, we're tackling the sample prep challenges. By providing various standard and control, we aim to fulfill the microbiologists' need for their study, such as assessing workflow bias from DNA extraction to library preparation, and accurate measuring microbial absolute abundance. Furthermore, we aim to help preserve microbial profiling and eliminate cold chain storage and transportation. With DNA RNA shield, we not only able to achieve those goals, but also preserve DNA fragment size for long read application. With DNA extraction and purification, we have achieved unbiased slices with optimal fragment size for HiFi sequencing, eliminate the need for addition, sonication, and cleanup. Just by using Voltec with horizontal adapter, which you can see on top of the Voltec right there, at max speed for 40 minutes, result in complete lysis of all organisms, but gentle enough to retain DNA length. Testing the fecal sample, we were able to achieve a fragment size upward 12 kb. In regard for library prep, we have optimized and customized the condition, such as primer design, PCR condition. The workflow is now sample type specific. With stringent QC protocol, Zymo service team can ensure the optimal result for individual project. And as important as sample prep, like I mentioned before, we continuously update our curated database and improve bioinformatic pipeline to get the most accurate analysis. So for now, you can enjoy all these perks of this optimized state-of-the-art workflow from collection to conclusion at the price of 16 short read. By simply send us your sample, we will handle all the work and hand you back a personalized data analysis report. Here's some information that you can use to actually reach out to us. We are gladly to help provide any service needed. Thank you very much.
Great, thank you, Ethan. Again, exciting to see the increase in reads that you're seeing at your site. And again, it's my pleasure to introduce our next speaker, Carolyn Howard, who comes to us from the Tree of Life program at the Wellcome Sanger Institute. And she's going to talk about a lot of the exciting updates and milestones that they've been able to achieve as some of the thought leaders in the biodiversity space. So with that, I'll hand it over to you, Carolyn.
Thank you very much. It's very nice to see everyone, and thank you very much for the opportunity to talk about the work that we've been doing at the Wellcome Sanger Institute. My name is Caroline Howard. I lead the wet lab team as part of this program, and we do an awful lot of long-read sequencing for the purposes of reference-level genome assemblies. So at the Tree of Life, we are involved in several major reference genome sequencing projects. At the top there, you can see the Darwin Tree of Life project, which has the ambitious aim of sequencing 70,000 species that are found in Britain and Ireland. We're also involved in the aquatic symbiosis genomics project, and there we're looking to sequence both the host and the symbiont in one go, so we can understand more about those relationships. We're also involved in the VGP and the European Reference Genome Atlas, and all of these come under the Earth Biogenome Project. But the main reason that I'm showing you this slide today is to just give you a taste of the huge variety of samples that we are working with. And of course, we are across the whole breadth of eukaryotic diversity here. So the way that we achieve this is with our genome engine. The samples come into us, go through the sample acquisition process, and they follow this pipeline all the way through to reference genome assembly, and all of these data are made publicly available as soon as possible. So I'm here to tell you mainly about the process that's the DNA and the RNA extraction and all of the preparations that we do in order to get these amazing long-read sequencing that's required, along with the high-C library sequencing, in order to get these reference-level genome assemblies. So what have we done so far? We have done an awful lot of sequencing. So we have 5,040 species with sequence data. We've produced 124 terabases of data just from PacBio sequencing there. In assembly, we have 1,941 species, and the ones that have gone all the way through all of our processes and been publicly released, we now number 1,256 assemblies. So when we reached the 1,000 milestone, we were really proud, and we wanted to make sure that we'd published and shared all of the protocols to describe exactly how we had achieved this. So you can see there on the right-hand side that we're not just narrowing this down to one particular area. We do have a number of arthropods that have been sequenced, but the red lines that you can see on the outside of that plot, they represent the species that we have assembled and released. So you can see that they are interspersed all the way around this wheel. They include fungi, they include arthropods, cnidaria. Really, if it's eukaryotic, then I hope there will be something in there that's of use and interest to the community. The lines that you see in blue are the ones that we have in progress. So as you can see, we are hoping to really fill the whole circle as soon as we possibly can. So what is required in the lab to deliver the 1,000 genome milestone that we're so proud of? Well, we can make it really quite simple. We need to homogenize the samples. We need to do high molecular weight DNA extractions. We need to fragment the DNA, clean that up, and we need to do the RNA extraction. We then need to make sure that we produce all of the data forms that you see here in green, and all of those data then go through to our assembly team who put them together. So in the laboratory, from a really practical point of view, this is what we have done. So we've designed a suite of standardized protocols, and we work on these all the time, and we're adding new ideas and new publications to them. But what you can see here is all of the ones that we have so far made publicly available in Protocols.io. The QR code that's shown there should take you directly to those protocols. And they've been designed as a kind of mix and match. So for any sample that we need to process, we'll choose something from the green box at the top. That will direct how we prepare the sample and the type of disruption that we use. We then choose a method from the pink selection. That's our DNA extraction method. Take that through to the orange, where we're looking at how to fragment the DNA and how we'll clean that up. And in blue there, you can see our RNA extraction protocols. So I'll give it a little bit of an idea of each of those. But with this standardized and yet flexible approach, it's really allowed us to progress at pace, but also with consistency. So at each of these steps, we collect exactly the same QC metrics. We collect them for all of our samples, and all of these are stored in our electronic lab notebook. So at the very top of that process, we have three main methods of sample disruption that we use. For more delicate samples, we use the power masher that you can see in somebody's hands there, actually in Amy's hands in the lab. We use the Cavaris cryoprep for samples that need to be pulverized. And we also use the MP-Bio fast prep for bead beating. At the moment, that's mainly for plant samples, but we're seeing some great results expanding that out as well. Then move on to the DNA extraction protocols. As you can see, there are a number of these. We also have a number more that are working in the laboratory. They're just not quite at the point of being published on Protocols.io as yet. The ones that are shown in a darker color and in bold are the ones that we're currently working on. We've still made public our previous protocols, so you can see how we've changed and adapted over time. But we thought it was important to let you know exactly which ones we're using. Something that's really important here is that we do have a mix of manual and automated protocols. So we have the Kingfisher Apex shown there in the bottom left. That means that we're able to process 96 samples simultaneously, and we do still get that high molecular weight DNA. However, there are occasions where we do want to process samples manually, and that's really something that our experts in the laboratory will cast their eye over, and they will pick and choose the ones that they feel should be processed manually. I've given an example of a femto profile there. Of course, it's a very beautiful femto profile that I've shown. They do not all look like that every time. But that's really what we're aiming for, really high molecular weight DNA. You can see in the portion towards the bottom left, there's nothing there. There's no low molecular weight or degraded DNA. That's what we're aiming for every time. And then here we just have our fragmentation methods. So we tend to use the G-tube if we're aiming for an ultra-low input PacBio library. We find it quite convenient. And we use the megaruptors for the fragmentation for the HiFi libraries there. Then we have the SPRY process, which is cleaning up of that fragmented DNA, which is really crucially important. Removing anything that's low molecular weight at that point will really have a huge impact on your sequencing yield. Again, we have both a manual and an automated process for that. We do find that the automated SPRY is pretty much as good as a manual SPRY. And the people in the lab really prefer it because if anyone has practiced a SPRY, it's quite tough on the thumbs. We also have a manual and an automated RNA extraction process. They have both had an equal success rate. So again, we're happy with either, but the automated version is higher throughput. So here we are. This is what we're kind of confronted with. A sample comes in. We have all of these options. Now, what do we do? So for example, if we have a moth sample that comes into the laboratory, through our tried and tested methods and having processed a lot of samples, we know that this is our ideal combination for Lepidoptera. So we'll go through our sample prep and triage. We know that we'll take the head to send for high C. We'll use a power masher on the thorax, automated magatract extraction, and we would anticipate that the yield and quality of that DNA will go straight through to a HiFi library prep, automated SPRY, and we would be looking easily to finish that genome in one sequencing event. So how about a grass sample? Well, we know that we need to beat the samples, plant samples. They're very tough, they're quite recalcitrant. The bead beating is really great there. The automated plant magatract, because grass tend not to give us too many problems. So we would go through that process. And again, we would be anticipating a HiFi library there. Bivalves. So mollusks can be quite tricky. However, we have seen quite a lot of success with bivalves. So this would be our first pass. We would cryoprep the material. We would be looking for the automated magatract. And we would hope that we would get enough yield and quality DNA for a HiFi library there. However, I would say we have about a 50-50 success rate in that type of a sample. So failing that, we would move on to the manual mollusk nanobind extraction process. So we do find that ones that fail through that routine magatract process, for some of them, we are able to achieve what we need using the nanobind. Fungi samples. Fungi is actually proves to be quite a lot tougher than you might have expected initially. So actually the bead beating protocol has shown a lot of success with the fungi. We've been able to increase yields using the bead beating in comparison to cryoprep. We use the plant magatract extraction. And for fungi, we do tend to preferentially go for the ULI libraries there. We find that fungal DNA doesn't always sequence the way that you would like it to sequence. You might have a library that looks great, but then the yield of sequencing isn't as you'd expect it to be, and we think it's probably to do with some of the unusual things that happen with fungi DNA. However, we find that the amplification in the ULI library prep helps us to overcome some of that, and genome sizes tend to be quite small, so we can multiplex those. And then a slightly more challenging plant sample here. So orchids, we do find, again, are quite difficult to deal with sometimes. We would go for the bead beating and then a plant organic extraction method that has been developed within our laboratory. It's a kind of semi-automated process, so it does have a phase separation, but then we do move on to the kingfisher with a bead purification of the DNA. Okay, so having all of these protocols and having these standardized methods does allow us to then work at pretty high throughput and go through at scale. So here you can see the PacBio sequencing that's been happening over at the Sanger Institute. All of the blue dots are from our Revios, and all of the green dots are from our Sequel2s. So for a while now, we've been working to have maybe 60 sequencing events per week. We have decreased the number slightly with the Revio because we get so much more output per cell. So you can see there that we do have some really high yields. We have had a few that are really pretty much 100 gig from one cell on the Revio, but we still do have the variation. And what I really would like to stress is not everything works every time. So yes, we do have some failures, but we also have a large number of huge successes. Having put through this many samples and having this scaled approach, it does mean that we're able to push some of the boundaries. And of course, we do need to push some of those boundaries. So when we've been looking at our yield data over time, we do find that yield is not strictly dependent on the N50. So you can see here that although there is, you can see that when we have a longer N50, we do get great yields, but you can still see that in the middle there, we might have a shorter N50 and still able to get over 75 gigs. So we do find that there's room for maneuver. But why would we want to do that? Why would we not just load a perfect sample every time? Well, here's a great example of why we might want to do that. So Amy, who works on the chordates in our lab, she's processed a lot of samples from strandings or from events where animals have sadly died. And the samples that we have, we then have the opportunity to collect are from, sadly, necrosis. So these are very challenging samples to source, and we need to make really great use of these samples that have come to us in this slightly unfortunate way. This is an example of something that Amy would choose to process manually. And you can see on the bottom left there the femto profile of the DNA. So this is not a beautiful femto profile. It's 9.2 kilobase average fragment length. We did get a decent amount of it, though, 4 micrograms, so that's pretty good. So what we actually did was put this DNA directly into library prep without any shearing at all. And you can see on the right-hand side, we were able to run that library 3 times, and then we created another library for the 4th run there. And the yields that we've been able to achieve, this is from a Sequel2, so the 19 to 20 gigabase yield per cell from DNA of this quality, which, this is poor quality. So we were really pleased with these results, and we were able to assemble a full reference genome there. Okay, so a scaled approach does require specialist knowledge, and we have to have that balance between a kind of routine throughput and also being able to go in and cherry pick things. Again, when we look back at our data, we find that yield is not strictly dependent on clades. So you can see on this diagram here, we have the chordates in blue and other animals in yellow, plants in green. It's very difficult to find a trend in those data. We do see that they are highly, highly variable. And within each of the groups, challenging species remain, challenging samples remain. So there's no one-size-fits-all here. So a few examples of how we have managed to overcome some of those failures. Here's an example here from Ben, who works on plants. You can see on the left-hand side, one of our kind of standard extractions, 645 nanograms of DNA. That's a very poor profile. But Ben had worked with that and developed his plant organic extraction method. And then you can see on the right-hand side some very nice high molecular weight DNA, over 2.5 micrograms there. And again, he was then able to put that forward for library prep and sequencing, yielding 28 gigabases from one sequel2 cell. Another example of a very challenging sample are some of the lichens. So Priyanka has been working on these. And you can see in the progression of femtotraces there, on the left-hand side, a power mash sample, 14 milligrams of tissue input and a very, very poor profile. Incorporating the bead beating, you see in the next profile along, she was able to increase the yield. It's still not huge, and the quality is still not fantastic. However, it was enough to create quite a nice-looking profile for a sample to go forward for a ULI library prep. And there we achieved an 18 gig yield, which again was enough to complete this genome and get the coverage that was required. So another thing that we've needed to play with and needed to push the boundaries on is the OPLC, so the loading concentrations. And again, you can see here, as we pull down the loading concentrations, the blue dots are again for the Revio. We can find that loading as low as 50 picomolar there, we are able sometimes to achieve 75 gigs yield. So it doesn't happen every time. You can also see blue dots down there that go much, much lower. However, it does start to show us some of the possibilities. If we're in a difficult situation, we don't have many samples, we've only got one shot, it's often worth a try. So again, why would we do that? Well, here are a couple of examples where we did take the chance, we did load low, and these are two genomes that, again, we were able to complete, and fantastic yields that we were able to achieve. Both of these are on the sequel2. And actually, being able to complete these genomes when the samples have gone so far through our pipelines, it's worth taking that risk, and these are just a couple of examples of where it's paid off, and we do have many others. So then optimizing data output is really important as we are trying to assemble these beautiful quality reference level genomes for lots of different species. Some of the more challenging ones are the very small organisms with relatively large genome sizes. So isopods are a group that have challenged us. An example here, this is a very tiny organism and the genome is nearly 3 gigs. Today, we might achieve this on one run of the Revio. However, a little bit further backwards in time here, we split this DNA so that we could run one HiFi library and we ran 2 ULI libraries. This is from one DNA extraction, and it's a method that we're able to use to really maximize and optimize the amount of data output that we're able to achieve for a particular sample. Okay, so how this is really achieved, of course, is through collaboration and through the work of really dedicated individuals. I've put the QR code for the protocols up there again. What we would really like to do is collaborate with as many people as possible. We really want to share our protocols. We know that lots of people have reference genome initiatives. We really would like to talk to anybody who might be able to make use of our protocols. We would love for you to give us feedback on these. It's possible to do that on the protocols.io format, and we'd be really keen to hear from anyone who has successes or failures. And we also hold monthly R&D meetings, which are open to anybody. They're also recorded on Zoom, so if you're not able to attend in person, you can simply have the recording mailed to you afterwards. We take a different taxonomic group at a time. Each month we provide these reports. You can see an example in the bottom left there. We talk about all of our DNA extractions. We pull together all of our data, all of our metrics, and we provide them for anybody to take a look at. So, for example, if you are a PhD student and you are looking to start sequencing a particular kind of plant, come to our plant R&D session, take a look at our files, see if we've sequenced anything similar and how we were able to achieve DNA extraction. I know when I was studying, I would have been hugely appreciative of that, and I hope that it is helpful to lots of people. If you're interested, email the TOL admin people and just say that you'd like to be on the mailing list. Okay, so all of that work really does take a village, and I would very much like to thank all of our collaborators and our collectors. I've not mentioned them very much here, but of course we are hugely dependent on a huge network of collectors. There are a lot of teams within the Tree of Life who are very important to all of this work, and within the Sanger core facilities as well. SciOps are actually the people who run the sequencing machines, so we are forever grateful to those people. 2 very quick cheeky slides I'm going to put up. We do have 2 roles within the Tree of Life that are available. There's one for a senior bioinformatician in Mathieu's team, and another for a senior bioinformatician or software engineer in Shane McCarthy's team. The details are on the board, and Shane and Mathieu would be really keen to hear from anybody.
Great, thank you, Carolyn, and congrats again on the exciting milestones you've hit this year. So last but not least, I'm going to hand the presentation over to Josh Clevenger. He's got some really exciting data coming out of low-pass sequencing on long reads and also sharing updates of what they're doing at Hudson Alpha. So with that, feel free to take it away, Josh.
All right, this doesn't go high enough. It's okay. So it's a real honor to be here. Good afternoon, and thank you so much to Jake and PacBio for inviting me to speak about this. My PhD is in plant breeding, so I spent my PhD out in the field looking at plants and selecting them. But I'm very, very passionate about translating genomics for plant breeding. I believe very strongly in the power of using genomics to make that crop and animal improvement as fast and as good as we need to feed ourselves and have safe, healthy food for everybody. And I believe that there's always ways to make that better and to improve that. And so these are the fine human beings that grace me with allow me to work with them. I told them I wanted them to name the lab. I didn't want the lab to be the Clevenger Lab. That's my name. That's not what we do. And so they came up with the Genetic Innovation Lab. And so I wanted to mention that because as we get going, as I'm talking, you can understand where I'm coming from and what I'm talking about. We really are interested in taking these amazing genomic technologies that companies like PacBio provide for us and making those innovations to make real impact in the world. So when I was a PhD student, Wayne Parrott, who is a professor at University of Georgia, was drinking adult beverages with us. I had just developed a SNP chip for peanut, which I thought was the greatest thing anybody could ever do. And he said to me, well, Josh, in 5 years, what is the thing that's going to be better? And I said, could you please just let me enjoy this? I can't think of what else would I need? I just made a SNP chip. That's amazing, right? So I'm going to take you through a story of someone who's passionate about breeding a relatively small crop that's a polyploid that went through a really large genetic bottleneck and how we are leveraging these tools to make improvements. And so I'm starting 15 years ago. These brave souls came together in China and said, we're going to provide a genome for peanut, which at that time was ridiculous. You were not going to do that. And they convinced a lot of folks in the peanut industry to give money to them and that they were going to do it. And I started my PhD during this project around 2013. And seriously, the meetings were hilarious. It was like, what are we going to do, guys? We cannot use short reads to assemble this polyploid genome. This is not going to work. And not until the RS2, in the collaboration with the Hudson Alpha, were we able to assemble the cultivated peanut genome. And that took almost 10 years of money and work and meetings and hair falling out. And I like to talk about that because we need to remember just like how incredible that actually was. And a lot of you probably remember those genome consortia that you were a part of. And so just 2 years later, this same group of folks, different group of folks, some a little bit older, are in Senegal in a peanut field where 300 accessions of peanut had been genotyped and sequenced and spread all across the continent of Africa to 12 different countries and phenotyped in those countries. And all of those students and all of those breeders were able to take those genotypes and map traits that were interesting to them and look at how those peanut varieties actually did across 12 different countries on the continent. So having that genome opened up the world for all of these people that probably for a long time didn't think they'd ever be able to do something like that. And so what we do is we're really interested in accelerating the translation of genomics for plant and animal improvement. And that's all we really care about. That's what we want to do. And so having those genome resources, we're like, how do we genotype better, right? I developed 2 peanut SNP chips. They were both terrible. I hate them, and I don't like to use them. And folks that still use them, when they talk to me, they're like, I don't understand why you're telling me not to use this. This is the paper you did it. Well, it's because if you use SNP arrays, you know that there's lots of issues with ascertainment bias and not really being able to sample the variants that you want to have for the gene discovery that you're trying to do to make it impact, to make a real impact in your crop or your animal species. So we basically developed a way in the lab, an analytics tool to analyze low coverage sequencing fast, efficiently, in a reproducible way. We worked with different companies to automate high throughput library preparation, to do the low pass sequencing. And again, we're starting with short reads here, allowing that core mapping and filtering and calling genotypes, imputing them, mapping genes, and then integrating that into breeding programs. And so that's where we started 2 years ago. So it's kind of interesting that I'm talking to you here about when we get to the end, that 2 years ago, that's when we started this. So here's the peanut guy again. I bought this on Etsy, I think. I thought he was cute. So he's going to be up every time we're talking about peanut. And here are the groups across the world that we now get peanut seed samples or DNA sent to us, and we sequence them and give them genotypes back. So we work with INTA in Argentina. We work with the consortium of groundnut breeders across Africa that is sponsored by USAID and other folks. ICRISAT in India and Australia, and then various groups across the United States. So going back to the first slide, having that genome and those brave souls that spent 10 years with nothing until PacBio saved them, opened up all of this possibility for groups in other countries that can just mail something to us and we can give them high quality genotypes back. Even during war. So when the war first started in Israel and Palestine, samples were sent to us from that region because they do grow some peanuts there. So that's an incredible thought. So I want to give you an example of how powerful this could be. So this was a smut, peanut smut in Argentina, right? It's completely devastating. The spore takes over the seed. You can't see it until the end. It survives every kind of possible condition imaginable and can spread all over the world. So when this was first discovered in Argentina as being a problem, it essentially rerouted supply chains, right? So I used to work for Mars Wrigley, which produces candy bars and kind bars. They buy a lot of peanuts from Argentina. It completely disrupted the global supply chain. Those peanuts were supposed to go to Europe. They were supposed to go to Asia and other places. And they couldn't because they were barred from different countries because they didn't want to spread this really horrible disease all over. You also can't bring samples to the United States. You can't bring spores to the United States. We can't be testing for this disease in the United States. So how do we work on it? Well, if we can identify a very strong resistance gene to start integrating in breeding programs without having to test, we can actually start tackling this problem. So the breeder in Argentina, Jorge Baldassari, which at the time did not believe in any molecular breeding. He was just pulled in because Mars and the USDA wanted to push this and try and develop something. He said, all right, I'll try it. I said to him, we have this really great low-pass sequencing. I want you to put as many plants in your population out as you can and phenotype them all. So we put out 600 F2 plants. Phenotype them. So to phenotype for this, they actually have to open every peanut seed one by one and check for spores. It takes months to do. So actually breeding for this trait is completely awful, right? Because you have to put everything in a field that has a lot of disease. So in that first year, we used the low coverage sequencing and we identified an extremely strong locus. We then validated that locus, identified a gene and variation in the gene. Then we went back and looked at a diversity set, which is on the left. And this is a box plot of percent infection when you have the variant on the left, which some of you probably can't see it because it's basically 0. I didn't make that up. And then on the right is when you have the other allele. So it basically has a lot of smut infection. And not only that, but in those populations where we mapped, they were actually his breeding populations, which is what I am really excited about, integrating genomics in the breeding program where a way where you're discovering and selecting at the same time. And so because we had all of the sequence, right, all of the genotypes, we could say, these are the lines that are resistant and also are mostly like this great agronomic line. So you can start making crosses with those and like accelerating the breeding process. So you can start to think about all sorts of ways that you might integrate that. So at this time, we were sequencing a lot of genomes because I was at Hudson Alpha and we had Sequel 2s, and I was like, this is the most amazing technology that I could ever imagine. And then they made something better, but at that time. And so we said, how do we utilize this in a breeding program, right? It's cool to like make genomes and say, I did a plot, a synteny plot, and this is different. But for a breeder, I just need to know what the differences are, where the variation in the alleles in my population. So we put tools together that other people had developed, and we took our pipeline and we integrated it into a pangenomic space. So now when we have populations where we have resistant and we have susceptible parents, we're now mapping and identifying those alleles onto the pangenome. We have the ability to see those structural variants with the skim-seq very fast and effectively, and you can start really expanding this on how you might use it in breeding. Multi-parent populations, multiple phase genomes, and then my colleague Alex Harkis and his lab is really excited about looking at variation of sex chromosomes within species and populations. Lots of opportunity there. And so the other thing is, if I'm using skim-seq, and this actually was sent to me by my computational biologist yesterday, so I asked him, can I put it in here? If you're using skim-seq on a pangenome, you probably want to impute your missing data because it's such a good system that you can sequence very, very low. So you want to have a complete data set. Well, we developed imputation, so lots of people, other folks are going to do that as well, but our imputation in a pangenome is almost perfect, even at 90% missing data. So because you have this higher ability to understand the variation in your population, you also have a higher ability to impute the missing data in your population. So there's a lot of really high benefits. And then harkening back to the previous presentation, folks like you all who share your protocols across all of these different species, allowing us to build these pangenomes with the sequencing, that all sort of comes together. It also you will start seeing PhD students who are in their first and second year talking about how they have populations in peanut to look at leaf spot disease, and they're using pangenomes to identify genes directly from those populations, which when I did a master's degree with Esther Vandernap, maybe getting from 1 megabase down to 60 megabase was a PhD project, and that was 6 years. But now you can start to think about these PhD students are like doing these things because of the power that they have. Okay, so this is where the really fun part comes. So Peggy Ozias-Akins and Juliet Chu allow me to work with them as well. Together we schemed up this 16-parent magic population that incorporated all of the known really high value traits in peanut. So really strong resistances to key diseases, tolerance to drought, mitigation of aflatoxin, and then some elites at high yield grade and those other agronomic traits. We sequenced all 16 parents. And so in this population that took about 300 crosses to make, we have all the parents in a pangenome graph. So other than any sort of new mutations, we have access to every single variant in this population. And it's a great breeding population. It's so great that we started a breeding program that we call Hudson Alpha Wiregrass. And in this breeding program, because we can sequence and genotype effectively, we do it with the students in the area. So this area in the wiregrass, 60% of the peanuts are grown in the United States in a 50-mile radius. And so we go in and they each have a plant and they extract the DNA from those plants. We sequence them and analyze them, and then we talk about what a breeder might be looking for. We're tracking 6 to 8 really high-value traits in these populations, and because we're looking at these magic populations that have gone through all this crossing, there's some really excellent lines in there, and we have the ability to see all the variation because we are mapping to a pangenome graph. So each student now gets their own plant, their entire genome of their plant, and what we show them is, so the larger dots are structural variants, and the smaller ones are SNPs. We're basically telling them this is where the elite background is. So there's a couple that have elite background, and these are where these traits are in your plant. So they can look at, this is what my plant looks like. This is the genetic architecture of my plant. And then we talk about which ones are we going to take and cross and move on to the next generation. And so this is another example of what it looks like. And because it's such a powerful way to track alleles, we are actually being able to tag up to about 8 really beneficial alleles in this population. And we know each line that has those alleles go back to the parents and then understand exactly what the genomes look like in these lines. So as a breeder, when I was doing my PhD, that wasn't just, you might do, I'm going to do background markers 1 per chromosome arm, right? We're basically looking at, in peanut there's less, but we're basically looking at 50,000 markers per chromosome arm. Okay, so this is my son Phoenix. His name was Phoenix before Paris Hilton named her son Phoenix, I guarantee you that. So the question is, what if, right? So 2 years ago, Alex Harkis brought back this Lego kit for the sequel 2, and my son put together, and he was so proud. And I put that there because at that time, we didn't know they were about to hit us with this Revio machine. So when you see that throughput, you think, well, what if I genotype my populations with long reads? That sounds pretty exciting. Is it possible? Well, actually it is. And so Kendall Lee is a new postdoc in my lab, and she's right there. So if you'd like to talk to her about some of this, I'm going to embarrass her and point her out. Please do. So again, we have this population of peanuts that we've skim-seqed, right? We've analyzed. So on the bottom is the short reads of an individual. And then we took that same plant and we looked at long read low pass on that individual. So the top is the long read low pass. And what you're looking at here is when you are trying to identify SNPs with short reads, in my opinion, I do this a lot, but I could be wrong. In my opinion, the greatest source of variation is knowing where that read actually it should align in the genome, right? So that variation is giving you problems and false SNP calls and errors. So we do a lot of filtering out. So on the bottom, that's what we see with short reads. And then the top, that's the long read low pass. And we're able to tag an entire R gene that is segregating as an extra, extra gene in our population. So we know which lines have it and which lines don't have it. So that's a really powerful thing to be able to do as a breeder. So the next part was, okay, we know what we're going to see, but we anticipate what people might ask of us, which they've already asked of us when we talk about long read, low pass. What am I gaining? Because I'm going to pay a little more, or I can show you what you gain. So we think about confidence of where that read aligns when there's a SNP. So this is the short read data, the Illumina data, okay? And again, this is aligned to a pangenome graph, so it's not a linear genome, so you can talk about reference bias. And then we looked at the long reads and he said, how sure are we of the long reads, right? And Jake didn't start my timer, so I'm sorry if I go over time. But as the read gets longer, actually, as you'd expect, our confidence is around 93% at 13 kb, meaning that almost 100% of the SNPs that we call, we don't filter out because we're not confident in where that read is aligned. Not only that, but the average quality of the HiFi reads that we measured this is a Q40. So I'm not worried about the quality of my call. I can do some things to offset that. And so just having 1 or 2 long reads covering a SNP or structural variation, I can feel confident about calling that. So really very exciting. And the other part was, okay, are we just not going to get as many SNP calls as we would normally? And that's not the case, obviously. As the read length goes up, we're covering many more SNPs. In this case, this is peanut. Peanut only probably has 250 to 400,000 SNP variants between any 2 lines. So in a long read low pass situation, we're and around 13 kb, reliably genotyping about 220,000, so close to more than 80%. And so I'll leave you this one more sort of visual here. So that the absolute stunning ability to understand the alleles in your population with the short reads in the bottom, and again, it's sequenced to the same amount of coverage, it's just that because I'm looking at 150 base pairs, I actually have to filter out most of them. So in this region alone, which is about, I don't know how big this is, Kendall, how big is this region? 18 kb, there it is. In 18 kb, there's 9 SNPs, and I can only, in short reads, I can only see 1. So actually you might think, well, it doesn't really matter that much. Well, it kind of does, because if you're trying to find functional variation, if you're not trying to just find a SNP that is kind of linked to your trait of interest, but you're trying to find the causative variation, you want to have everything. So this is really, really, really exciting. So again, I'll leave you with this. The story is that where we came from and where we are at now, the idea of genotyping completely just with long reads is not only viable, but it's something that we should be doing. And so this is the funding support for the work that we did. And again, my biologist, Walid Karani and Kendall Lee are the ones that did most of the work that I presented you today. So thank you very much.
Thank you, Josh. Really exciting stuff. I would actually, if anybody has questions, we do have a few minutes. So apologies that you just sat down, but if I would like to invite speakers back up to the stage in case anybody has questions for our speakers today. And then if you do have questions, there's a microphone on each side, so feel free to just come up and we'll take them in the order that they arrive. So yeah, Ethan, Josh, is Carolyn still? There we go. And then Doreen. Perfect. So if anybody has questions, please feel free to come up. Perfect. Hey, Tim.
Hi. Is this on?
Is the mic on? If not,
Hello. Not working?
well, it didn't tell me. It is on. Okay.
So the question's for Doreen. With the Kinnex ISO-seq version, and since you have good data on both the Kinnex and SQL for similar samples, and I've asked Jonas this a couple times, but the more data we get, the better I'll feel. Have you looked specifically for any bias against longer transcripts based on the way that Kinnex works to ligate things together? So do you start missing things at the longer end of transcript length?
So I can say that we looked initially at the data. So as a person who's older and done a lot of PCR work in amplicon bias and concatenation, we actually looked preliminary, but I have no statistical analysis, to see if we were getting any bias of insertions of smaller pieces. And so I can say that there is a little bit more, not statistically, but we see some more smaller reads. But what I can tell you is there didn't seem to be a bias associated with the size of this transcript of the 65 loci that we identified, or 65% of the loci that we identified. So in the seedling sample, we were actually impressed that we ended up seeing 65% of the known genes identified. And it didn't seem like there was a bias because the gene was longer that we didn't see it. And in fact, there's sort of like an urban legend that things that are longer or have more introns in them are more likely to be highly expressed and things. So there are some bias. So I can tell you just qualitatively, we see maybe a more smaller inserts, but it didn't bias against us getting the long reads, the longer transcripts in the full-length data sets. And then I just wanted to clarify that
Okay.
I don't think there's a problem.
I have a question for Caroline about the work you've been doing. first, I think it's an amazing achievement. It must be a logistical nightmare, so congratulations. But I was just interested to know, from all of the assembly corrections that you've done, you said you've done quite a lot of manual interventions. Have you started noticing any patterns of things that you constantly have to fix? Because you're in a position where you've probably sequenced the most genomes now out of anybody. Is there any advice that you can give us of what to look out for?
Well, particularly in the assembly. Is that correct?
Yes. Yeah.
Well, I'm looking in the direction of Shane McCarthy over there, who deals with the assembly. I think he would probably answer that a lot better than me as to whether there's anything that you consistently come across, but he's just there.
Okay, no worries. I had one other question as well. I noticed that the yield for the Revio was somewhat broader than the previous machine. Is that because, does that mean that the machine is kind of a fly or die at the moment, or are you really going to get really good yield or really bad yield, or are you being quite risky with what libraries you put on? Is it because of the machine, or is it because of your libraries?
I would say it's likely because of the library, and it's probably because the proportion of the sequencing events that you could see on those graphs is kind of, obviously because of the timing of the release of the Revio, it's kind of clustered, and it includes all of our beta testing. So all of the validation runs that we did with, really pushing the boundaries to see exactly where we need to stop. So, okay. But yeah, we see the trajectory is moving upwards over time as well.
Thank you.
Thanks.
First, thank you for all the amazing talks. Yeah, I have a question for the last speaker. I want to ask you about your experience. Like you showed us amazing results about imputation using the pangenome. Is it only SNPs, or do you have experience in?
No, so that actually includes the structural variation, because if you treat them as alleles, you're imputing them as allele calls.
Oh, yeah.
So not just SNPs.
Thank you.
Great. If there are no more questions, let's give a round of applause to our speakers again for putting on an amazing workshop.